Dear participants, welcome all of you. Today, we will organize our first workshop in the scope of Buca Imsef 2022. Dr.Arzu Uyar is our keynote speaker with the topics of motion in the biomolecular world. We would like to give her a brief resume first. Arzu Uyar received her undergraduate degree from Istanbul University Department of Chemical Engineering. Afterwards, she completed her master's and doctorate degrees in Boğaziçi University Department of Chemical Engineering, working on computational biochemistry in Professor Pamela Dorpar's group. She carried out her postdoctoral studies in Professor Leonard Nilsson's group at Karolinska Institute in Stockholm for 1 year, and then Professor Alex Dixon's group at Michigan State University for 5 years. She has recently established her new team in Izmir Technological Institute Bioengineering Department and continues her studies in the field of computational biochemistry. Dear guests, we would like to invite Ms. Arzu Uyar to the stage for her speech. The stage is yours.
Thank you very much for this kind introduction, Nad. But first of all, what I would like to say is I would like to thank all the organizers, all the committee members in Buca Imsef Festival. I really enjoyed being there. I was not there, I know, but I really enjoyed actually to be with you. It was a great time, and I hope maybe next year I will be in the team again. And today, I would like to briefly talk about what we are working on, not only in our team actually, but also as a field like computational biochemistry, what we do. I would like to share like a brief information. So let me share my screen with you, and please let me know if you cannot see anything. And I'm gonna start my slides. Okay, I hope everything is clear right now. Okay, and I hope you can hear me. Can you hear me?
Yes, perfect.
Okay, all right, thank you. All right, so welcome to the motion in the biomolecular world. Today we're gonna visit this world, the biomolecular world. I'm actually an assistant professor in Izmir Institute of Technology in the Department of Bioengineering. And I actually named our group as Uyar Biomodeling Group, and very briefly, shortly, we call it U Biomodel, because you actually biomodel in this group. Okay, next slide. First, where we are in the world. We are in Turkey, and we say hi. How we say hello? We say merhaba. So merhaba, everyone. Hello from Turkey. We are here. Actually, we are in Izmir. Izmir Institute of Technology was established in 1992 in Izmir, on the west side of Turkey. And this is the Department of Bioengineering website. And this is our fantastic students and our professors, as you can see in this picture. And one thing I think which is very important about the Department of Bioengineering in ISTEC is all of the professors in this department, they in their academic career, they did research actually, not only in Turkey but also in other countries, other cities. So this is quite important. Okay, let's go back to the scientific part. So the motion in the biomolecular world, if you would like to understand the motion part, first we need to learn a little bit about the biomolecules. What are they? How can we visualize them, right? So in this picture, on the left, what you see is an amino acid. So which has an amino part, which is this part, and then the acidic part, as you can see here. And we have R1, which is the, we call it side chain, which makes each amino acid actually a unique one, okay? So on the left, we have amino acid 1 because the side chain named as R1. And on the right, we have another amino acid 2, and the side chain is named as R2. So these two amino acids, they come together, and then a water molecule, as you can see here, a water molecule releases, okay? And then a very strong peptide bond actually gets formed, okay? So there are 20 different common amino acids. So that's why actually each protein structure is different, is very unique actually. So here in these tiny circles, you see different types of amino acids. So I will not explain all of these, but there is a chart key here. So as you can see, some of them are aliphatic, aromatic, acidic, or basic, or hydroxylic, or some of them might have a sulfur atom. All right, and they have, each of them has actually a different name. Like this one, the first one is alanine, and we don't have to write alanine all the time. We can just type actually A, and we know that it's alanine. And another example might be proline here. And when we say P, we know that this is the proline actually. It is called like a one-letter code. And here there is also 3-letter codes under each name, like pro here. This is the 3-letter code. I mentioned that some of them might have a sulfur atom. So for instance, this one, cysteine, which 1-letter code is C. This one has a sulfur atom here, as you can see here, which is S. Okay, so we have different types of amino acids, and actually they come together. As you can see on top of the slide, it says primary sequence. It looks like a necklace. I wish I wore a necklace today, but I didn't. So this looks like a necklace, right? And each bead actually here represents a different amino acid. They come together, and then they form a secondary structure, as you can see on the right-hand side of the slide. The pinkish color here represents a shape which is called beta sheet. And this one, the blue color represents here an alpha helix. Totally a different structure, right? So they don't look like each other. And you see a tiny loopy structure here, which actually connects these alpha helices or alpha helices and beta sheets. And this is called secondary structure. And then our protein actually get folds, and it makes its tertiary structure, okay? But finally, if our protein would like to function, actually perform its function, then it might need its quaternary structure. So this is the quaternary structure. Okay, now you might ask, if you haven't seen any proteins before, now you might ask, do all proteins, do all these macromolecules look same or quite different, as you can see in these vegetables or fruits, okay? So some look like this, like a pear, and this one looks like, this is a tomato, right? And tomato and apple, actually, they look like a little bit similar, but again, they are different, right? Actually, how we get that information? Some scientists, they are called experimental researchers or experimentalists. So they do lots of experiments. So they crystallize protein structures, 3D structures. And these structures are actually kept in the protein data bank. It is actually a bank, but it's free. It's open to everyone. So I highly recommend you to go online and check rcsp.org website, as you can see here, rcsp.org. It is very useful, and they also have learn section here where you can learn a lot of things about protein structures. Even after today's seminar, you can go, or right now you can go and check it out, what this RCSP website is. And they also tell every month they pick like the molecule of the month. So the November molecule of the month looks like this. Okay, it says like actin branching by ARP2-3 complex. Okay, so this is where we get actually our 3D structures, protein structures. So now I would like to share a couple of different structures with you. This is the structure of red liver walt. And I forgot to mention that each protein structure has an ID like us, right? Like humans. So this one has a PDB ID, as you can see here. So in this one, in this figure, actually, we see the top view of the structure from the outside, actually. And it has how many amino acids they came together and the water molecule releases and the peptide bonds formed here, like almost 32,000 amino acids formed here. I mean, they came together and formed the peptide bonds and etc. And in this structure, we have almost 242,000 atoms. It's a huge system. And now we see the same structure from different angle, and this is the inside view, right? So each different color here actually represents a different unit of the structure. And the front view would look like this. Isn't it a huge molecule? Okay, another one, human apolipoprotein A1. So I really like this one because if you look at from this angle, it looked like the number 8. And on the right-hand side, if you look at from that angle, it actually looks like number 0, right? And this one has 804 amino acids, and in total 6,600 atoms. So this one is a little bit less than the other huge molecule. Another example here, antifreeze protein from the beetle. And again, this one has another PDB ID, as you can see here. So on the left-hand side, what we see, 2 different colors, right? So 2 different colors here means 2 different units, okay? When there is only 1 unit, we call it's a monomeric structure. It's a monomeric protein. When we see 2 different colors here, that means there are 2 units, and we call it a dimer structure. It could be a homodimer or heterodimer. Why it could be a homodimer? If these 2 monomers, if they look like same in terms of the sequence, if they are exactly the same, then we call it a homodimer structure. But it could be a different sequence here and a different 3D structure, then this would be a heterodimer. So on the right-hand side, we see the same protein, same antifreeze protein from the beetle, but we are looking at from a different angle. And it looks like this. It seems like there are like big holes here, big 2 holes. And this one actually is a small structure because it only has 164 amino acids and in total 1,100 atoms. Okay, another example here, clathrin. This one is again a little bit a big one because it has 15,300 amino acids. It's a really big one. Okay, how about viruses? Due to the COVID-19, we got used to all the COVID or other virus-related news recently, right? Or for the last 2 years, actually. And here you see different types of viruses. I just wanted to share this with you to show you like viruses also have different kinds of shapes. Their 3D structures are different. Okay, as you might notice, I shared all these tiny pictures with you. Then now you might ask, are these molecules, these macromolecules static? Actually not, right? Atoms wiggle and jiggle, okay? So welcome to the Protein Dynamics show. And the structure of a protein will have some dynamics, and actually it will take us to its function. Here, you see a tiny video. Actually, our atoms wiggle and jiggle. I hope you're able to see the tiny video here. some atoms, they come closer. Okay, now we have another animation from inside cell, a very short animation, this one. But you see all these molecules, actually, they are dynamic molecules. Okay, couple of years ago, researchers from Japan, the Institute RIKEN, and from United States, from Michigan State University, they actually, they were able to simulate, they were able to model the bacterial cytoplasm. This was a huge, huge research. The reason for that, I hope you can clearly see here how many different macromolecules are in this system. And there are also water molecules in the system. If we zoom in, we see lots of water molecules. We have a couple of different ions here. We have metabolites. So we have all kinds of molecules in this in the system. And this one is a full atomistic system. I want to take your attention, actually. So now I'm going to show you a short video from that system, how this system actually behaves. Okay, so the dynamics of the system in atomistic detail. Maybe it's not very clear from that picture, but what we can do, we can zoom in, we can focus on one region, right? So let's do that. Just a second. Okay, let's focus on one region and see how these molecules interact with each other. So now we have different proteins here, actually. Like the yellow one is a different protein. We see a red one, we see orangey one or dark gray one. And in the middle, we have another molecule, another macromolecule, and all interact with each other. You see how this part, this alpha helical part, actually opened and then got closer to the other structure, which is in red color. So all these computational biochemistry tools actually will show you this kind of dynamics. Okay. Then I would like to share with you another example. This is called unbinding dynamics. What I'm trying to say actually by saying unbinding, assume that, okay, we get sick, right? Most of the time, unfortunately, and then we need to take some medicine, right? We need to take some tiny drug molecules. And actually, those drug molecules, they go and they bind to their target, all right, to actually inhibit or probably activate the function of that protein. So we have on the right-hand side, you see the one protein here, and not in the middle, but as you can see here in blue color, we have a tiny drug molecule. So then the drug molecule does its function, right? And then it needs to actually exit the structure, leaves the structure. So this is the video of how a drug molecule or a candidate drug molecule actually leaves the structure. You see two important loops here. They probably open, so the drug might enter, and so then they get closed. And now you see, so this was the drug in the middle of the figure, and the other molecules around it from the protein, those were actually the amino acids. Actually, here you see the 48 different copies, but you don't have to pay attention to that. So in the end of the video, you will be able to see not all of the 48 drug molecules would leave, actually, but some of them would leave the structure. Have you noticed? Some are already started leaving the structure. And there we go. Okay. So some of them actually exit the binding site. All right. So those were like really fancy calculations, fancy simulations, I know. And you might have this question like, how can I calculate the dynamics of a protein? Is it possible? Can I do it at home? Okay, actually, you probably need supercomputers. You don't always need it, but you would need supercomputers, like high performing computer power. Okay. In the figure, this looks really fancy, right? All these blue lights and etc. But this is from probably a movie. This is a movie scene probably. But in reality, this would look like this. So this is a supercomputer at Michigan State University. So we took that picture like a couple of years ago, and you see lots of cables and things. But actually, this is a supercomputer. It was one of the powerful computers in the US, actually. Okay, so what about the curious researchers at home like you? So because I know that you might not have an access to a supercomputer. So is there anything you can do? Actually, the answer is yes. And actually, I have two different answers, which is a good news, I guess, right? So the option one would be Google Colab. All right. Here, actually, I'm sharing with you one of our simulation results where actually we performed the simulations on Google Colab account. So what we did, we opened the Google Colab account, and then we mounted our Google Drive, and then we have all the input files, like the protein data bank files from the protein data bank website. And so we set up our system and then perform the molecular dynamics simulations using different tools. I didn't want to provide actually lots of detail about all these molecular dynamics simulation programs, but if you are interested in, please let me know. You can always reach out to me. I will be happy to share those kind of details with you. And actually here you see a water, all these red, I would like to go back. I don't want to miss. And here, what you see here, this is an enzyme. And inside the enzyme, we have two different extra molecules. And our enzyme is in the middle of a water box. So this is how we simulate actually our simulations. So why we put water molecules? What is the reason for that? I know some people already have an answer. Because we mimic the cell environment. And we know that in the cell environment, we have water, right? So we mimic the water, we mimic the cell environment by using the water molecules. And if our protein is a membrane-bound protein, then what we would do, we would definitely add the lipid bilayer to mimic the cell membrane. But this time, we don't need a cell membrane, we only need water molecules. And this is now I'm showing all of the amino acids in a sticks representation where you fully see every single atom in the system. And the dynamics already started. You see all these atoms wiggle and jiggle. And this is called a cartoon representation. So both our protein structure actually mobile right now, and also the ligands inside the enzyme, it's also mobile. Okay, so this is the Colab website. Why I recommend this? It's a free one. That's why you could use Google Colab. All you need is to have a Gmail account. All right, then you can start using the Google Colab to perform your own MD simulation, which is free. But of course, it has some limitations, but it's better than nothing, right? Okay, we have another option, option 2. It's called Elastic Network Models. Or ENMs. in this case, what we do, we don't use all of the atoms, and we don't use the like mimicking environment, cell mimicking environment, the water molecules. We don't use that, which is called the coarse-grained model. All right. This is our protein. We don't take into account all of the atoms, but this is a cartoon representation of our structure. So we can clearly see all the helices and these beta sheets and all these loops, right? Then we only take into account alpha carbons. So please try to remember the amino acid structure. We have C carbon atoms, we have nitrogens, we might have some sulfur atoms, right? Or oxygens, of course, and hydrogens. But this time we only take into account the C alpha, carbon alpha atoms. And then we draw a hypothetical shell actually, or a sphere actually, which has a radius here, as you can see here. And if the distance between two C alpha atoms, as you can see here, for instance, this one and this one, if the distance between these two is within the cutoff distance we determined, then we say, oh, these two atoms or these two C alpha atoms are in contact. Then we generate our network, 3D network, elastic, like this, or it's called elastic network. This is an elastic network. Then we do a little bit calculation. So there is mathematics, there is definitely physics in it. We do a little bit calculation, and we are able to actually get these kind of motions without performing molecular dynamics simulations. So in this case, this is another type of enzyme, another type of protein called neurolysine. And one of its motions, it's called like hinge bending type motion. So you can assume it like this. So it does this kind of motion. It's opening and closing actually like that. And the one on the right-hand side, this is called scissor-like motion. Now we are actually looking at the structure not from this angle, now from it's like a side view, and it does this kind of motion, like scissors. And actually, these kind of motions tell us lots of information about its function. All right, next one. And finally, I would like to introduce you our group members, because without them, I wouldn't be able to perform any simulations or any modeling, right? I won't be able to do that by myself. But currently, I have lots of students. I'm really actually lucky since I have them in my group. And actually, some of our group members, I couldn't get their pictures. I'm so sorry about that. So we have many other members in the group. I would like to thank them for being in the UBiomodel group. And if you have any questions, I would be happy to answer. But actually, before closing, I would like to share a website with you where you can actually perform elastic network modeling, if I still have time. Do I still have time? No?
Yes, absolutely.
Okay. Let me very shortly go to that website. All right. Okay. Just a second. First, I would like to introduce Professor Yvette Bahar, because this is her website from the University of Pittsburgh. So Yvette Bahar and Burak Erman, Professor Burak Erman, they were the They actually created the elastic network model. And I really like the Yvette Bahar's website. So every time I go to the software and resources part and to the A&M part, which is the anisotropic network model, and then you see this anisotropic network model web server, which is so easy to use. You just need to enter the PDB ID of your protein, or you can just submit your own protein. So this time we can use 4-AKE. This one is adenylate kinase. Actually, this is like the hydrogen atom of biology. And in this time I will write A, so we're going to use only one chain. And then click calculate. We just need to wait a couple of seconds, sometimes couple of minutes. It depends on the structure, size of the structure. But the 4-AKE has 214 amino acids, so it's a small one. And then in this website, now you can see the molecular motions actually of this protein, of this enzyme. You see the mode types here. There are different modes. That means there are different motions. So this is the number one. You can increase or decrease the frequency of the mode. You can play with the amplitude scaling, or you can actually turn on the vectors and actually increase the length of the vector to better visualize what's happening here. So in this case, the adenylate kinase structure, it has 3 important regions. They are called domains, but it's okay to call them a region right now. So one region, one domain, which is called the lead domain here on top of the structure. And here in the middle of the structure, this one is called core domain. So this is the core region of the protein. And on the right-hand side, this part is the NMP binding domain. Or the region. So what we see here, actually, we see lead domain and NMP domain. They are actually come closer. So the protein, if we say the protein is an open mode, it's open right now, it's actually doing this kind of motion, right? Opening and closing type of motion. I'm sure you noticed that this is a harmonic type of motion, right? It's an harmonic, so opening, closing, or doing the scissor-like type motion. So this is the first mode. You might ask why we have different colors here, because this part is red and this part seems like a little bit pale blue, and this is almost whitish. The reason for that, the lead domain, the top part actually moving, or the dynamics of that part is actually bigger compared to the other part of the structure. So this is more flexible region. We can call it like that. This is more flexible, this region. That's why we have actually longer vectors, as you might notice, compared to the core region, right? So this was the mode 1. Let's see what mode 2 looks like. And in mode 2, we lost the vectors, but I'm gonna reactivate it. Okay, in the mode 2, actually it is doing that kind of motion. Now the lead domain is not closing towards the NMP binding domain, but what I see, it's going toward the right, to the right actually. This is what it's doing. And you can generate your own actually motion. So you can generate your PDB files, protein data bank files from this website. You can download all of the results from this website. I know it is too much, but I think it's full of fun to pick one molecule from protein data bank and upload into the A&M web server and then see what kind of motions your protein would perform actually. I think that was all, and okay. And I would like to thank again for every single person who attended and all the members who organized this nice meeting.
Thank you so much.
Yeah, if you have any questions, I would be happy to answer.
We can take questions from the chat part, or you guys can join via voice, or we'll also take questions from the YouTube live stream. If you have any questions, don't hesitate to ask.
Actually, I forgot to say one more thing. If you would like to, okay, I'm a computational biochemist, so even our group meetings are mostly online. So if you would like to join our group meeting one day, we'll be very happy to host you, actually. So please email me. Do not hesitate to email me. Just email me and tell me, hey, I would like to attend your group meeting. It doesn't matter you are a professor or a student or you're not a student. It's totally okay. We'll be happy to have you.
Dr.Arzu, Uyar, thank you so much for your speech. It was really informative and enjoyable for us. I would like to ask you that, do you have any advice for the young scientists in Buca Imsef who want to study protein dynamics or bioinformatics?
Mm-hmm. Okay. Okay, they should immediately start learning how to code. Yeah, programming. Any kind of programming language is okay. It doesn't have to be Python. When I was a master's student and PhD student, I used to run my write and run my codes in Fortran programming language, and I guess I don't use it anymore. But I still love Fortran, to be honest. Or I used to run my codes in MATLAB, which is not free, so it might be hard to reach out to MATLAB program. But Python is free, or R is free. So I highly recommend people start learning Python or R currently. And another actually advice would be, please do not hesitate to contact people. Okay, this is very, very important. Don't be shy. You might have questions. Just email that person, or in Twitter or LinkedIn, I don't know, any kind of social media platform, okay? Just go online, try to find that person. If that person doesn't reply to you, then try to find another one. This is what I can actually advise.
Do we have any other questions? I'm also checking from YouTube live stream if we have questions. If you guys would like to ask questions, you can use the voice, or you can just text on the chat.
Or maybe now everybody is checking the ANN web server, either Protein Data Bank website.
Okay, looks like we don't have any further questions. So thank you one more time for your great presentation. Our workshop is over. Thanks for everyone's participation, and goodbye to you. We have another workshop tomorrow, and then on Friday, we have our closing ceremony and award ceremony. So see you guys then.
Okay.
Thank you so much.
Thank you.